Have you ever marveled at the sheer complexity of a human being? Consider the intricate network of nerves that carry our thoughts, the powerful muscles that allow us to move the delicate tissues that form our senses. We are, each of us, a walking, thinking, feeling miracle of biological engineering, and yet, this extraordinary creation begins in the simplest way imaginable. It starts not as a grand design, but as a single solitary cell. How can one microscopic entity contain all the instructions, all the potential to construct a person? This question is one of the most profound in nature, a mystery that unfolds in the quiet, hidden world of the womb. It is a story of division, migration, transformation. The journey from a single cell to a fully formed organism is perhaps the most remarkable feat in nature. The moment of conception marks the beginning of this incredible saga. A single fertilized egg, the zygote, is formed. It is a tiny world unto itself, complete and self-contained, holding all the genetic potential for the individual it will become. Almost immediately it begins a process of rapid cell division known as cleavage. The first division creates 2 cells, then 4, then 8, then 16. With each division the cells, now called blastomeres, become smaller. They huddle together, forming a compact ball that looks much like a miniature mulberry. This structure, known as the morula, is the first collective achievement of these new cells, a tiny solid cluster making its slow and steady journey towards the uterus, its first home. As the cell count climbs past 32, a remarkable transformation begins to occur. A fluid-filled cavity, the blastocoel, starts to form within the center of the cellular mass. The resulting structure is the blastocyst, a hollow sphere with 2 distinct cellular populations. On the outside is a single layer of cells called the trophoblast. Nestled inside the blastocyst at one pole of the sphere is a distinct cluster of cells known as the inner cell mass. It is from this inner cell mass that every single tissue, every organ, and every bone of the future person will arise. Once the blastocyst has successfully implanted in the wall of the uterus, the inner cell mass undergoes its next great transformation. This process, called gastrulation, is a marvel of cellular choreography. The cells of the inner cell mass migrate and reorganize into 3 distinct layers, stacked one upon the other. These are the primary germ layers, the ancestors of every single cell in the adult body: the ectoderm, the mesoderm, and the endoderm. From these 3 simple sheets of cells, all the complexity of the human form will be sculpted. The outermost layer is the ectoderm, meaning outside skin. This layer is fated to form the boundary between the new individual and the outside world. But its responsibility extends far beyond simple covering. A portion of the ectoderm will fold inwards to create the entire nervous system. The brain, the spinal cord, and the vast network of nerves that permeate our body all have their origins in this outer germ layer. The middle layer, nestled between the other 2, is the mesoderm, or middle skin. This layer is the source of our structure, our power, and our internal transport systems. From the mesoderm develops all of our muscle tissue, from the mighty biceps to the tirelessly beating heart. It forms our skeleton, the strong and flexible framework of bone and cartilage. Finally, we have the innermost layer, the endoderm, meaning inner skin. This layer folds to form a simple tube that runs the length of the embryo, the primitive gut. From this humble tube will develop the entire lining of the digestive tract, from the esophagus to the intestines. Furthermore, the endoderm forms the lining of the respiratory system, including the lungs and airways, creating the internal passages and glands that process the food we eat and the air we breathe. With the 3 germ layers established, a new and deeper question arises. How does a cell from the mesoderm know to become a heart muscle cell and not a bone cell? How does an ectodermal cell know to form a neuron instead of a skin cell? The answer lies in a process of extraordinary subtlety and precision called cell differentiation. It is directed by a complex and beautiful symphony of molecular signals that guide each cell towards its unique destiny. This guidance system works through a constant conversation between cells. Cells release chemical signals like tiny molecular messengers that travel to their neighbors. These external signals ultimately work by influencing the cell's own genetic library, its DNA. Within the nucleus of every cell is the complete set of genes for the entire organism. However, in a differentiated cell, only a specific subset of these genes is active or expressed. The signals a cell receives activate special proteins called transcription factors, which act like keys. They switch certain genes on and others off. This process is a cascade of commitment. Early on, a cell's fate is quite flexible. An early mesoderm cell, for example, has the potential to become many different things. But as it receives more and more specific signals, its options narrow. It becomes progressively more committed to a particular path, until it reaches its final, terminally differentiated state. As cells differentiate, they do not remain as isolated individuals. Instead, they begin to cooperate, organizing themselves into communities of similar cells. These organized communities are known as tissues. From the vast array of specialized cells, the body constructs just 4 fundamental types of tissue. Epithelial tissue is the great liner and cover of the body. Epithelial cells are packed tightly together, forming continuous sheets. They cover the body's surface as skin. They line internal cavities like the digestive tract and airways. They act as a protective barrier, controlling what enters and leaves the body. Connective tissue is the most diverse of the 4 types. It connects other tissues, supports other tissues, and binds other tissues together. This tissue type includes tendons, ligaments, cartilage, bone and blood, cells suspended in plasma, connecting the body through a vast transport network. Muscle tissue, also from the mesoderm, is defined by its ability to contract, to generate force, and to produce movement. Its cells are elongated and filled with specialized proteins that slide past one another. Cardiac muscle, which forms the wall of the heart. Lastly, nervous tissue derived from the ectoderm, is specialized for communication. It consists of neurons and glial cells. Together they form the body's rapid information network. The brain, the spinal cord, and the nerves. Cells do not build tissues in a vacuum. They exist within a complex 3-dimensional environment that they themselves create. This intricate network of proteins and carbohydrates secreted by the cells is known as the extracellular matrix, or ECM. It is far more than just passive packing material or cellular glue. It is an active and essential component of every tissue, providing structural support and vital information. The composition of the ECM varies enormously from one tissue to another. In bone, for example, the matrix is hard and rigid, fortified with calcium crystals to provide immense strength. In contrast, the matrix of cartilage is firm but flexible, rich in water-trapping molecules that make it an excellent shock absorber. In connective tissue under the skin, the matrix is a gel-like substance, filled with strong, rope-like collagen fibers for tensile strength, and stretchy elastin fibers for elasticity. The ECM is not merely a physical scaffold, it is also a communications hub. It stores and presents a variety of signaling molecules that can influence cell behavior. The physical properties of the matrix itself, its stiffness or softness, can also send signals to the cells within it. A cell can feel its surroundings, and this mechanical information can profoundly influence its destiny. During embryonic development, the extracellular matrix plays a dynamic and critical role. It provides pathways for migrating cells, acting like a roadmap that guides them to their correct destinations during the formation of organs. The interaction between cells and their matrix is a continuous dialogue, a partnership that sculpts the developing embryo. It is, in a way, the interplay between the cellular actors and their stage. Once the primary tissues are formed, the embryo enters the next grand phase of development, organogenesis. This is when tissues organize into functional organs, folding, splitting, clustering, hollowing, transforming layers into a 3-dimensional body. The development of the nervous system is one of the earliest and most dramatic events. A section of the ectoderm on the back of the embryo thickens to form the neural plate. The edges of this plate then begin to rise like 2 ridges, forming the neural folds. These folds curve towards each other and fuse in the middle to create the neural tube. This hollow tube is the precursor to the entire central nervous system. The front part balloons out and folds to become the brain, the rest elongates to form the spinal cord. At the same time, within the mesoderm, the first stirrings of the circulatory system can be seen. 2 simple endothelial tubes form. They fuse into a single, primitive heart tube. The tube begins to twitch, then to beat rhythmically. It pumps primitive blood cells through a rudimentary network of vessels, even before chambers form. The tube elongates, twists, folds back on itself in cardiac looping. It gradually partitions into the 4 chambers of the adult heart. Meanwhile, the endoderm is undergoing its own transformations. The primitive gut tube formed by folding of the endodermal layer begins to specialize along its length. Regions become the esophagus, stomach, intestines. Small buds grow outwards, branching into the liver, gallbladder, pancreas. At the front end, an outpouching forms. It bifurcates and branches repeatedly, creating a tree-like structure of the airways and lungs, gradually assembling the complete set of tools the body will need to survive in the outside world. Birth is not the end of the story of tissue development, it is merely a transition. The processes of growth, maturation, and repair continue throughout our lives, from infancy to old age. Tissues must grow as we do, they must be maintained and repaired in the face of daily wear and tear. A scraped knee, a broken bone, the renewal of the lining of our gut all depend on cell division and differentiation. The architectural work may be largely complete, but cellular maintenance workers stay on duty for life. The key to this lifelong capacity is small, hidden populations of remarkable cells called adult stem cells. Found in many tissues, bone marrow, skin, gut, they are unspecialized, but when they divide they can self-renew, or produce a cell that will differentiate and replace damaged cells. In skin, stem cells produce new cells that migrate up and replace shed surface cells. This regeneration echoes embryonic development. After a bone fracture, nearby stem cells activate. They form cartilage-forming cells and bone-forming cells, create a bridging callus and eventually remodel into solid bone. In the liver, even if a large portion is removed, the remaining cells can be stimulated to divide, regenerate the lost tissue, and restore function. This innate healing ability is a vital feature of life and a testament to embryonic design. Studying tissue repair is not just academic, it promises new medicine. Understanding stem cell signals could let scientists harness healing to treat many conditions. Regenerative medicine aims to repair or replace tissues and organs, targeting heart disease, diabetes, spinal cord injury, Parkinson's disease. The dream: instruct the body's cells, or use lab-grown cells to rebuild what was lost to injury or disease, recapitulating embryonic creativity to heal the adult. As we trace the journey from a single cell to a complex community of trillions, we are witnessing one of the most beautiful and profound narratives in all of biology. It is a story that unfolds according to a set of rules written into our very DNA, a biological script refined over eons of evolutionary time. The intricate dance of cell division, the fateful migration of cellular tribes, the whispered conversations that lead to differentiation, all of these steps reveal a system of breathtaking elegance and precision. To understand embryonic development is to glimpse the fundamental mechanisms by which life builds itself, a process that is at once overwhelmingly complex and beautifully simple. This knowledge does more than just satisfy our curiosity about our own origins. It provides a foundational understanding of the living world. The same basic principles of tissue formation, organogenesis, are at play across the animal kingdom, connecting us to the vast web of life on our planet. These are ancient processes, a common heritage that reminds us of our place within the grand tapestry of nature. Furthermore, this journey into our past holds the key to our future health. Every disease at its core is a disruption of normal cellular and tissue function. By understanding how healthy tissues are built, we gain invaluable insight into what goes wrong during illness and how we might intervene. The study of embryonic growth is the bedrock upon which the future of regenerative medicine is being built. The hope is that by learning nature's secrets of creation, we can learn to become masters of recreation, persuading our own bodies to heal in ways we once thought impossible, mending hearts, regenerating nerves, restoring sight. Ultimately, the story of how a single cell becomes you is a source of profound wonder and inspiration. It is a reminder that within the smallest, simplest beginnings lies the potential for extraordinary complexity and beauty. From the quiet darkness of the womb emerges a thinking, feeling, living being, assembled with a precision that surpasses any human craft. And in understanding this miracle of construction, we not only uncover the secrets of our past, but also find a bright and shining hope for the future of human healing.